Thank you, and good morning, everyone. Day two of a really interesting summit, as is always for these WEF meetings. We're going to get started today with a 1 hour panel. This is going to be with a number of speakers. They're going to highlight some of the ideas and thoughts from their wastewater surveillance programs. And we're going to go one by one. I'll introduce them. I'm not going to go too detailed into their bios. That's available in the digital program. And then we're going to have plenty of time for questions at the end. So please reserve your questions for the Q&A. We will, of course, have virtual participation online through Zoom as well. So our first speaker for today is Dr. Doris Dye. She is the lead wastewater microbiologist at the Hawaii State Department of Health, and she oversees their lab's wastewater monitoring program. Doris is going to be presenting virtually. So Doris, if you can, yeah, those are your slides. You can go ahead and get started.
Hello, good morning. I hope everyone can hear me.
Yes, we can hear
Yes.
you.
Okay, thank you so much. Good morning. Thank you for inviting me to present today about wastewater research priorities in Hawaii. So I'll go ahead and start with some brief introduction. Next slide, please. So in Hawaii, the wastewater surveillance program started in year 2022. And since then, we have weekly testing and biweekly reporting. So we report all the data that we have from news, from wastewater scan, and as well as from the state lab here in Hawaii. So in total, there were 9 wastewater treatment plants participating in the wastewater surveillance program. These 9 wastewater treatment plants serve around 69% of the total population in the state of Hawaii. So we consider this pretty high, despite we still have a lot of cesspool, septic tank in locally. And we also have another project with the long-term care facilities. For now, we have 4 long-term care facilities that we do routine testing for the wastewater. And this program started 2023, and we have been achieving very good results. We were able to cut some of the outbreak in all of these facilities. Next slide, please. So what are we testing? We started this program with COVID, and then we added more target in our routine testing. We added RSV, influenza A, influenza B, and then last year, since our first detection of the high pathogenic avian influenza in the wastewater, we started subtyping H5 concurrently with influenza A. But since now the season has passed, so we stop subtyping concurrently, and we only subtype H5 when we detect influenza A. So later in the year, when influenza A started spiking again in the wastewater, we add it on to our routine testing again. And we also test for norovirus. While this virus is abundant in the wastewater, as we all know. And other than that, we are developing some other assays for some emerging pathogens that we're interested in. First of all, it's Candida auris and also carbapenem-resistant genes and bacteria. Next slide, please. How do we test it? So over the years, we compare and tested many different methods. For sample collections, for the long-term care facilities, we are currently comparing 2 different methods, auto-sampler and passive sampler. And concentration, we started with InnovaPrep concentrating pipette, and then comparing with the nanoparticles, and then now we're using the series nanoparticles for concentration and extraction because it depends on how many volume of the downstream assays that you need. So we did compare of many different kits, and we found that the OPrep Power Viral is more suitable in our assays and workflow. For quantification, we also compare different methods, including CFA Gene Expert and digital PCR. We want to compare which method is more more effective and shorter turnaround time, but also more sensitive. So right now we're using digital PCR. Next slide, please. So for targets and pathogen prioritization, in my opinion, these are the 5 points that I think is relevant and important. First of all, would be the public health relevance. Depending on the endemic and emerging diseases in your local state or jurisdiction. For example, in Hawaii, pathogens that are commonly increasingly reported in Hawaii will be leptospiral and also salmonella. And also some of the outbreak history in your local state. In Hawaii, we have some outbreak of dengue and hepatitis A back in the days. And clinical burden, depending on what are spurring around in your location. High incident in the clinical settings is also important in determining the target and pathogens for wastewater surveillance. The next one will be environmental and geographical relevance. The rainfall and stormwater runoff is one of the key points, and also sewage infrastructure vulnerability. For example, some of the aging infrastructure has some leakage and then rainwater or stormwater will be able to seep in and bring in some environmental contamination. Tourism hotspot is important key in Hawaii because we have a lot of tourists. So you might want to think about whether you want to do wastewater surveillance in the tourism hotspot to determine what are some of the pathogens that are related to travel. And feasibility and methodological considerations. This is very important in the lab perspective because we want to know whether these pathogens are detectable in the wastewater and stability in wastewater. Specifically preference of pathogens and genes for non-environmental persistence. So you want to know whether some of the viruses circling around your community were able to be detected in the wastewater. How long are they gonna be stable in the wastewater, and will they be degraded over time and stuff like that. And sample metrics compatibility. Regulatory and programmatic priorities. This one, we think this is a very important factor as well because you have to be aligned with the news priorities or our local Department of Health priorities. And one of the important key factor of this is also the funding, whether we have enough funding to do additional target detection or some development. Population impact. For me, this is a very important factor in our local communities. Some of the local communities have limited access to healthcare. So wastewater surveillance in this community would be very important, and it serves as a very valuable tool that we're able to know whether they are impacted by some of the emerging or endemic diseases. And of course, we are doing the nursing home wastewater surveillance because this community has high chances of getting infected, and because of their health uh, condition, so we want to help them with wastewater surveillance as I mentioned earlier, we have some successful detection of the outbreak in this, and we are able to help them and control the spread among the facilities. Next, please. So I think all of these are very important because it will emphasize the need of collaboration between the EPI and the lab. Next, please. So I want to talk about some environmental contamination possibility. So one of the pilot project that we did over the years, and we're currently still doing it, is the AMR detection in wastewater. And we specifically interested in carbapenem resistant genes in the wastewater. So the first graph that I show here are the clinical cases in the state of Hawaii over the years. So as you can see, KPC and NDM has been increasing throughout the years. And we are worried about this because we don't get a lot of detection from previous years, but recently it has increased. Next, please. So we did some wastewater testing using digital PCR. So using the method I mentioned earlier, we want to know whether this carbapenem-resistant pathogen has been uh, circulating in the community for a long time, or it just started to come out. So from the wastewater result, you can see that KPC and oxal has been detected constantly throughout the sampling time at a high concentration. NDM is always detected as well, but we get some IMP detection over the time. But comparing to the clinical data, we rarely see any IMP infection in the clinical settings. So this makes us think whether this detection were because of the biofilm in the pipe or from the environmental contamination to the sewage or something like that. So next slide, please. So these are our questions when we did the pilot project, whether it is from human, from the environmental, or from the biofilm. Next, please. So we don't really know, and we still don't have the answer for this. I think wastewater surveillance will be a tool, be able to help with this, but we still need a lot of research, still need a lot of answers and questions to be able to apply this for EPI to take the actions in helping the community for this outbreak. Next, please. So I'd like to acknowledge some of my team members for their hard work in the wastewater surveillance program. Thank you.
Thank you, Doris, for your presentation. We're going to move to our next panelist. I'd like to introduce Dr. Prabhu Gounder. He is the medical epidemiologist at the Los Angeles County Department of Public Health. He works to undertake surveillance and outbreak investigations and is the lead for the CDC News program in the LA County.
All right, good morning, everyone. So today I wanted to focus on some of the not advancing for me here. I'm trying to figure out why. Should I be using this? Is that? Oh, you're advancing. Sorry. Okay. Good to know. All right. So actually, yeah. I'm ready for the next slide. So I want to focus on some of the key questions that we would need to answer to be able to translate, wastewater testing data into public health action. And so these are, questions familiar with all of you. First of all, can the pathogen be detected? And then if so, what is the minimum incidence that it represents when you have a detection? And what is the best approach for then analyzing and presenting those results? Do these results then correlate with their available clinical surveillance data? And if not, then to try to understand why they don't correlate. What are the reasons for that discordance? And then how does this add to what we know from other sources of surveillance? Kind of answering the question, so what, that we often get for wastewater. And then ultimately, what is the best way to communicate these results to policymakers and the public? Yeah, this is pretty basic and intuitive, but I thought it would be helpful to sort of break this down into distinct steps and then look at a couple of examples for how we would approach this, starting with SARS-CoV-2, where we have a lot of experience, and then talking about hepatitis A, which is a new pathogen for us. So next slide. So with SARS-CoV-2, we knew early on that it was detected, which is great. But that's not the case for a lot of pathogens. I'm thinking about the ones that we're monitoring locally. In LA County, we've got thousands of cases of C. auris, but very few wastewater detections. Early on with measles, my understanding was that it was, maybe an earlier version was an assay that didn't separate between vaccine type from wild type virus. And we know people are getting vaccinated all the time, and we weren't detecting it in the wastewater. So what does that mean? I think the newer assay is potentially more sensitive, but before, when we had cases, we didn't have wastewater detections. Thinking about dengue, we have about 100 to 150 travel-related dengue cases a year. 90% are DNB3, but what we detect in wastewater is DNB1 and 4. We have, I don't think ever had a detection of DNB3. So again, this idea of detection shouldn't be assumed, and I think that's the starting point for translating any wastewater to public health. But once you do that, then the next step is to see how well it correlates with our clinical data. And this is just an example done by Angela and others in the room here, showing that actually there's a really strong correlation between clinical reported cases and wastewater concentration. So that made us confident in the data because, again, early on, whenever you get wastewater data, people are hesitant to trust it immediately. It goes up, it goes down. Sometimes it doesn't correlate with what you see clinically. And so people aren't sure, can you trust this data? So this is an example of the type of work that would be necessary to lay the foundation of trust for policymakers to feel comfortable using it. Next slide. And then we have to, once we see the correlation, think about how do we present it? Because a lot of times we're looking at the raw data, but the raw data is messy. There's a lot of variability. And so this is an example of a situation that we confronted during the pandemic. This is looking at a 2-year, 2 years of data for the raw measurements of SARS-CoV-2. And, I highlighted a couple of outliers that are clearly outliers in red, but it's not always easy to know what an outlier is. And I circled a couple in blue where they're above the baseline, but it's not clear that it's an outlier. And those are examples where we had to make a decision because we were using an indicator of a % change over 21 days as an indicator of concern. And if you included those points, it actually changed the trajectory, the 21-day trajectory. And so we didn't want to feel like we're making subjective calls on something that should be objective. And so that's when, actually, next slide. We had to think about then the different ways of accounting for that variability. We tried a weekly average, we tried 7-day versus 10-day rolling averages, and ultimately what we settled on was a 10-day rolling trimmed average. So usually we get 5 measurements within 10 days, we dropped the highest and the lowest, and we took an average, and that rolling average seemed to correlate the best with our data. But you can see, again, it's not obvious. It looks intuitive now, but it took a little bit of trial and error to get to that point. But the main trade-off with that type of an approach is, when you think about the main advantage of wastewater testing, people say it's that lead time. People are going to be shedding before they even have symptoms and go and see a doctor. But if you have to wait 10 days to know that you have a reliable trend, you kind of lose some of that lead time advantage. So again, there's trade-offs in how we think about this data as well. Next slide. And then, well, first, once we are comfortable with the correlation and how we want to analyze it, then we got to think about how to present it. As I mentioned, on the upper left, we were initially looking at a 21-day change, but then this led to complications. There's times where you have really high levels of transmission, but then it's declining. So our indicator would say it's of low concern because the trend is heading downwards, even though there's just a lot of transmission. Others, like SCAN, have come up with the matrix where they look at the concentration, the wastewater concentration level, and the 21-day trend, and then come up with the wastewater category. But our director, thinking about, again, the end user giving press conferences every day, thought this was too complicated. Like, how am I going to convey this to someone that's easily understood? And so we ended up with what you see on the bottom, which is we pegged our level of concern to a percentage of the peak from the last year. And that felt intuitive to our director because people, especially during the pandemic, remember these surges and they can think back to the last winter surge and know when transmission was that high, what the EDs look like, what the hospitals look like, and what type of control measures they had to take. So then they can peg their actions to something that they were able to remember. So that's ultimately something that worked for us. Next slide. And then after all that work was done, it took 2 years to show the added value of wastewater testing. This is something familiar with all of you because I'm sure you've all seen this in your jurisdiction. But until then, wastewater was one of other indicators, and we still had really good PCR testing, and that's primarily what we relied on: PCR testing, hospitalizations, and death. And it wasn't until the Omicron surge and the second half of that Omicron surge in the summer of 2022, our director was looking at the PCR data and said, the case counts aren't that high, but the EDs are again getting impacted. Hospitals are again full. We're having a ton of outbreaks in nursing homes. And so we're able to then look at the wastewater data and overlay it. And this is around the time that the at-home antigen tests became much more widely available. And we're able to directly infer based on this type of a figure that actually you're right. Because of at-home testing, are people not potentially testing at all? we're missing about 40, 50, 60% of that community transmission. And so this is something, this was actually a slide from the press release that, the press briefing that she did on July 28th of 2022, where she was able to use this to say to the public, the transmission is worse than it appears. Please take it seriously. Next slide. So again, that was SARS-CoV-2 an example, and now we're going through all the same questions again with this outbreak of hepatitis A that I mentioned yesterday. Since last year, we've had about 170 cases of hepatitis A. This is an epi curve of those cases broken out by travel versus non-travel related, and the distinction being if it's not travel related, it's potentially locally acquired. So one of the benefits of wastewater testing, of course, is that area in gray that I have at the end. It shows an area that's underreporting. It really does take a lot of time to investigate and confirm a case, up to 2 to 4 weeks sometimes. So it is definitely underreported. And then underneath it, you have the wastewater concentrations, which are definitely much more timely, but you see there's a lot of variability. Next slide. And so some of the work that we're doing is starting with the beginning. So before the outbreak took off, what you see here is wastewater testing data with bars from one of the wastewater treatment plants that we received data from. The aqua are detections, the yellow are non-detects. And what I want to highlight are just a couple of areas of discordance. So in the arrow there, you see a case which we assume to have a 3-week shedding period where there's almost no detections. But on the other hand, you have a box which shows sustained detections but no case. And we're going through all the same questions again that we did with SARS-CoV-2. What are the reasons for this discordance between clinical cases and wastewater testing results? What's the limited detection of hepatitis A? How many cases do you need before you even have a single detection? What's the duration of shedding? How does that? And that's really becomes important because what I didn't show is once you start to get sustained detections, this type of non-detect is not helpful, and you need to understand how the duration and magnitude of shedding compares to your ability to, with what you see in the wastewater, for your ability to correlate. And then ultimately, how do we interpret and convey this information to policymakers and the public? And there's a lot of work that goes into it. So what we're doing now is going back to each of those clinical cases where there's a discordance and trying to figure out, did we misclassify the case because there's a lot of alternative reasons for hepatitis? Did we catch them on recovery? So maybe we just placed them at the wrong point on the infection timeline. So if you moved it over, maybe you would actually have discordance. Maybe, again, each case would require that type of evaluation to then better understand the source of discordance before you can then do the correlation, before you can then make policymakers confident in using it. So again, these are basic questions that I'm sure all of you intuitively know, but I just wanted to kind of tangibly show with a couple examples what that looks like, and we need to replicate this over every reportable pathogen. So at least in LA County, working with Zymo to develop sequencing approaches similar to what's done with TEFI. But once you get all that data, there's a lot more work to be done on the epi side for those correlations before we can start using it for public health.
Thank you, Prabhu. We're going to move to our next speaker. Our next speaker is Dr. Kirsten St. She is the Director of Virology and the Chief of Viral Diseases at the Wadsworth Center and a clinical professor of biomedical sciences at the University of Albany.
Thank you very much. Thank you for the invitation to join the session this morning. For the sake of time, I'm not going to go into a lot of detail about the program in New York, but just to say we started the wastewater program in March of 2020. And we cover approximately 200 sheds across the state and more than 85% of the population. And we have a pretty diverse program now covering many pathogens and a few other markers. I didn't join the wastewater program until 2022. It was being handled primarily by a commercial testing company, but we have been in transition now for about the last 9 months, moving the program from commercial into the Wadsworth Center. And I was primarily charged with planning and overseeing the initial stages of that. We recently brought in another director to take over and take care of the processing labs for the wastewater program. So it's been a big effort to transfer that whole program across to the state, and we're almost there. So that's sort of my history. So I'm relatively new to it, just since 2022, not from the onset, but it's proved enormously beneficial. So we have a large, diverse program. If I could have the next slide, please. Just have to make disclosures. I do receive research support from Thermo Fisher, Biorad, Kaigen, and Ceres, and I have royalty-generating collaborative agreements with Zeptometrics and MRN Diagnostics, and those things are relevant to the work that I'm presenting today. Thank you. Next slide, please. So I'm just going to touch on a few aspects of sequence analysis as we use it on wastewater and as we are proposing to use it on wastewater. Sequence analysis in wastewater is really becoming a very, very large field. This is not in any way a comprehensive presentation, but just a few highlights of some of the ways that we're looking at using it. So first of all, looking at RSV, had a big interest in RSV for a long time, but particularly the last couple of years, it's been particularly severe, and not just in children and elderly, but we've seen severe infections in other adult age groups as well. And of course, recently the approval of additional monoclonal antibody therapies and vaccines by the FDA, which has been tremendous clinically, but caused us to be somewhat concerned. Can we turn this sound up? Can I get closer to the mic? Is that better? Terrific. Okay. Okay. Is that better? Can you hear me? Okay. And so it also brings, with the implementation of any of these, either vaccines or monoclonal antibody therapies, comes a concern that, of course, the virus will start to mutate and generate mutations that will evade. We've had a couple of, we have a couple of substantial clinical surveillance programs running both in Albany, in New York, and with our collaborators in Rochester in the West, monitoring clinical samples for those evolutions, for those mutations. And we have already sequenced several hundred samples. But obviously, we get a much more comprehensive spread of data if we sample wastewater. And so we now also have programs initiated for sequencing RSV out of wastewater to look for those same mutations. And of course, the focus is primarily on the fusion gene for looking at that. And we are generating data as quickly as possible on those samples. We have a lot of stored wastewater material, stored nucleic acid that we can look at retrospectively to get that data pretty quickly. So that's one area that we are looking at. If I could have the next slide, please. Influenza has been relatively recently implemented across the country for wastewater testing in many sites. And I'm not going to show you all the data that we have. We started testing in January, but we went back to September stored samples. So our data is from September 2024 on. We implemented testing for influenza A, influenza B, H1, H3, and H5, all simultaneously, and RSV. So what we're looking at here is a heat map from September, on the left, September 2024, across to the end of March 2025. On the left-hand y-axis is a list of all the sewer sheds that we were testing in. We did not do all 200. We right-sized, as they say, down to a total of 87, focusing on high population centers, dairy farms, high level points of travel importations, and major hospital locations, and a few other sites of interest. So, and on the right-hand side are the sort of regions that those sewer sheds are in. I realize that you can't see the actual names. That's not the point of the slide. The point of the slide is to show this heat map going temporally from left to right. And the gray boxes are untested sites, the white boxes are negative, and the orange boxes are positive hits in the wastewater with intensity of color increasing levels of virus. And so you can see a significant seasonal trend as we go from left to right across that period of time, not surprisingly getting increasing positivity in the wastewater with hepatitis A, and increasing strength of signal as we come into the influenza season. We see, I'm not going to show all the heat maps, but we see, just, you have to take my word for it, we see a very similar picture with the H1 and the H3 and a somewhat similar picture, but not so strongly seasonal for influenza B. That tended to be a lot more even across the period. If I could have the next slide, please. This is the picture that we see with H5. So this is not really seasonal at all. It's sporadic across the entire period, and it is notably positive quite frequently. And there are certainly some sites that are positive more often than others, but we were very surprised at how often it's positive across the state, especially given that we have never had any positive cows. We've certainly had many positive birds. We've never had any positive cows, no positive outbreaks in any of the dairy centres, and certainly no positive human cases either. So this amount of positivity in the wastewater was somewhat of a surprise to us. This data has not been published yet. It's in the process of being written up for publication. It has been presented a couple of places. It was presented obviously to the senior level executives in the Department of Health. It's also been presented to the CDC, but you are the first other people to see it. If I could have the next slide. So as I said, the influenza A signals showed a strongly seasonal temporal pattern, but in contrast, H5 showed this sporadic positivity across the entire period with absolutely no seasonality and generally low viral loads, but nevertheless very consistent, with some locations demonstrating more frequent positivity. And we are in the process of developing H5 sequencing methods to look more closely at those positive signals in the wastewater, and we're comparing the sequence in the wastewater with the positive sequences in animals across the state. This is a collaborative project with Cornell, who've done a lot of sequencing of H5 positive animals, also in collaboration with New York City and their positive H5 wastewater, and Cornell again, comparing the H5 sequences in the wastewater there with the positive H5 sequences in live bird markets. So very interested to see if we can line up the sequence data in the specific areas across the state with the sequences that we're seeing in the animals in those exact same locations at different times. If we could have the next slide, please. So just touching on a few other areas and not going into a lot of detail or a lot of graphics either, but hepatitis A, we've had a long interest in hepatitis A. We have not suffered the same extent of outbreaks as some other centres, notably here in California, but nevertheless, we undertook an initial pilot study that was quite small in area, but long in duration, looking at Hep A across just 4 counties. But outside of New York City, we don't actually get many cases of hepatitis A. So we're now doing a collaborative study with New York City and testing their wastewater for Hep A and also sequencing the Hep A in the wastewater. For quite some time, we have done whole genome sequencing of hepatitis A, as well as short sequence to get the genotype. And we're correlating the case sequence data with the wastewater sequence data. And the interesting thing is that not only are we seeing some spikes of positivity in wastewater when we have no correlating cases, but we're seeing sequences of Hep A, whole genome sequence of Hep A, that is not always correlating with the sequences that we get for the cases that we have. So Hep A is a reportable disease in New York City and New York State, and we're fairly confident that it is well reported. And yet we are seeing a significant number of sequences in wastewater that are distinctly identifiably different to the ones we're seeing in the cases. And so we have an investigation underway to find out what are the differences in the cases that are secreting into the wastewater that we are not seeing in the case population that's being reported. We have some possible suggestions. Maybe it's pediatric cases that are asymptomatic, that have a different strain circulating amongst the children. There are some other possibilities that we're looking into there. But it's certainly an interesting exercise. Polio, of course, we had an outbreak of polio in New York in 2022, and we've been monitoring the wastewater ever since. We have not had positive wastewater since early of 2023, and we were about to decrease the amount of monitoring that we do. We've only been doing it in the areas around where the case occurred and the regions of the state where there are particularly low vaccination levels. And we were going to reduce, not stop, but reduce the amount of testing that we did. But just recently, we made the decision to not only not reduce it, but to actually increase it. And the reason for that is, one, the increased number of cases of polio being reported around the globe. Two, the ongoing decrease in vaccination coverage due to vaccine hesitancy in the population. And three, that we're heading into a summer season, and summer, of course, is polio season. And so we're actually going to increase the amount of polio monitoring that we do. One of the issues with polio, of course, is that when you get a positive hit, you want to correlate the sequence with the biggest database that you can, which is the international database, to see if you can figure out where it came from. And that is a very complicated exercise because access to the international polio sequence database is very restricted. So that is one of the greatest difficulties with polio sequence identification. We're also interested in enterovirus D68. We've had a long-standing interest in that virus, and we have a collaboration with 2 academic centers on correlating sequence with neurological disorders in children. And so we're interested in looking at the D68 in water for other reasons as well, for clinical triggers, but also to look to see if we can see if there are changes across time that correlate with the decreased incidence of neurological ramifications of D68 in children. And lastly, of course, there's metagenomics, which is a huge field with tremendous potential. Some people have said to me, what do you think you're going to do with the metagenomics data? And my answer is always, we're not sure, but we'd like to see what's there. And until we see what's there, we're not always sure what we're going to do with it, but that doesn't make it not worth looking for. And I owe Mark Johnson a tremendous apology because I was very excited about his project and I didn't have the bandwidth to get involved with it yet, but I promise him we will get there. I think that's all I have. If I could have the next slide. I have to acknowledge my tremendous team at Wadsworth and the other people within the Department of Health that are listed on the left and on the right-hand side, all of our academic collaborators and Quadrant Biosciences, who was the commercial lab that initiated all the testing for New York State. And at the bottom, always, our utility staff in the wastewater treatment plants, without whom absolutely none of this would happen. Thanks very much.
So thank you to all our speakers from today. We're going to move into a Q&A session. So if you have any questions, please feel free to speak into the mic so that our online audience can hear as well. We will be taking questions from our virtual audience. So if I can please request Prabhu and Kirsten to please come up. And we have Doris online. Any questions in the room? I want to give you a chance to ask your questions first. Otherwise, I do have some questions that we can kick us off with. Please go ahead.
Yeah, very nice. Just a general comment that speaks to your last talk, Kirsten. Because we do the sequencing program in the state of Texas every week on samples from 40 sites, and I have to say that I don't think there's a virus for which we have good sequencing data deposited in public repositories for which in the wastewater we don't see novel mutations or SNPs. There seems to be changes in a lot of these viruses, especially flu, SARS-CoV-2. You mentioned enterovirus D68 and many others, H5N1 too, when we were seeing that, that we don't see for sequences that are publicly available. So it just kind of speaks to that there's a lot of viral changes that are in the virus sphere in wherever these viruses are coming from that are making themselves in wastewater, for which we don't quite understand what those changes mean. So really appreciate the sequencing approach to that.
Sure. So we know that wastewater is giving us a much more comprehensive picture, right, of what's circulating in the community. The only thing that's getting into sequence, and I shouldn't say only because it's certainly a lot of data, but what's getting into previously, what's getting into this databases is from patients who are presenting clinically, which is for some diseases, a fraction of what's actually circulating. I mean, the extreme of that is polio, where 70% of infections are asymptomatic, and 30%, almost 30%, are indistinguishable from other viruses, and less than 1% are paralyzed. So there's a fraction of data in there unless you look at wastewater. Measles, on the other hand, is the other end of the spectrum. The vast majority, more than 95%, present clinically and frequently severely. So, and there's everything in between. But the point is that, unless they're presenting medically and they have a specimen collected and that specimen gets sequenced, it's not ending up in a sequence database. So it's a fraction of what's actually circulating. Whereas from wastewater, we're getting everything. And it's a much more comprehensive picture. And so I think what wastewater shows us is that there's a heck of a lot more going on in virus diversity, in genomic diversity in viruses, should I say, than we would be led to believe from the previous database conglomerate. Yeah. Thanks.
Do we have any online questions?
Oh, there we go. Okay, so we do have an online question, which Kirsten, I think you kind of just answered, but why are polio sequences so limited? And second part to that is, what advice do you all have for academics working with public health for their research in this area?
I assume the question means why is the access to polio sequences so limited? And the answer is that I'm not sure. You would have to that question would need to be directed to the keepers of the keys to the database, which is not even the CDC. It's, I believe, it locked up by the WHO. Martin, do you have any more on that? No. Is there anyone else in the room that knows who's locked? Yeah, somebody's got their hand up over there. Who's got the keepers of the keys to the polio database, and why is it so heavily locked up?
Hi, I'm Mami Taniuchi from University of Virginia. I'm one of the polio researchers in academia, but I have worked in the past with CDC and other folks. We do a fair amount of polio sequencing and wastewater stuff in Pakistan and other places. But we also have access issues to the database. It is held by the CDC actually, and the GPLN network, and it's not open for everyone to use, but there are investigators that you could partner with who have access to those data. So if anybody is interested, let me know. I can introduce you to some of those people.
All right, thank you. So that's good to know, but I have to say that even when we had positive cases and positive wastewater, we were not given access. So it's not easy, is the best
No,
I can
it's not.
say.
Yeah.
Yeah. You have to have strong reason for wanting to access those sequences. Yeah. Even if you have positive material.
Would you like to add to the second part of the question about advice for academic partners?
So I don't know who you partner with to be able to do that. I'm sorry, because we were not successful in getting access.
Prabhu or Doris, any thoughts?
There we go. Okay. I think that I really like the question about how to partner with academic entities, because as I mentioned, there's a lot of work to do. And I think one of the challenges for us in public health is we don't have the time to do it. We're oftentimes responding to, doing the case investigations, the contact management, the outbreak management. So, and especially with wastewater, we're still at the beginning, and there's so many pathogens, there's so much work to do at every step of the way. But at the part that I talked about in terms of translating the wastewater data to clinical practice, I'd love to partner with academic entities who might have the ability to support some of that analysis. I think the challenge is we don't have any funding to offer, But I think what we do offer is opportunities for people in training to get access to a data set and do the analysis. So I think it could be a win-win situation. And I think what that requires is establishing data use agreements with your health departments to be able to get access to both the sequencing data and the individual level clinical data that you would need to be able to do that type of work. But I think I definitely work. We work locally with our academic institutions, but encourage more of that type of partnership for data sharing to be able to spread the work for the analysis that needs to be done.
Doris, did you have anything to add?
Yes, I second what Prabhu mentioned in the earlier. We really don't have time to do some research or to measure what are the priorities, what are the targets that we don't want to add on for wastewater surveillance. So in Hawaii, we currently do not work with any academic partners, but we do rely on them for their research publication. So every time we want to add on a new targets in the wastewater surveillance, we do a lot of research referring to their published papers and understanding the sciences. And we also do journal clubs every month just to add on to our knowledge of what are the research, what are the techniques that we're advancing in the science, and what can we utilize in our wastewater surveillance locally.
I'm sorry, my previous answer was referring to polio. So we actually have a strong research initiative at Wadsworth and always have. It goes back with a long history. And so we have partnered for many years with many different academic collaborators. The list that I had at the end of my slide presentation, a number of those were academic centers that are part of the sequencing consortium in New York State, which I'm the scientific director of. So they partner with us for that, but they also join us with partnering for some other research initiatives as well. Our partnering with those springs from not so much from the wastewater program per se, but because elsewhere in the laboratory we have a lot of interactions with them from the clinical testing side of our testing program within the Department of Health. And so we have a lot of interaction with them from our clinical testing program, from our surveillance testing program, and so on. And we springboard from that into the wastewater program. And there's just a lot of crosstalk and building of relationships with investigators at various academic centers. And from there, I guess it grows from there. So there are some long-established relationships, but a lot of networking at conferences in informal settings as well as formal meetings and so on. So it's something that builds up over time. Thanks.
You had a follow-up question? No, I don't think.
What they think a sequencing-based dashboard may look like, a public-facing dashboard. This is something that we develop, but because of the complexity of information, it's difficult to translate that into something that's useful for either folks or itself. So just from both the metagenomic standpoint, health purpose standpoint, any key features that you guys once one
Before you answer, I'm just going to repeat that for the virtual audience. So the question is about thoughts on a sequencing dashboard and approaches to providing data to the public through that.
So it depends on who if you're talking about really to the public, then I think that's somewhat difficult because you're targeting an audience that may have very little, if any, technical knowledge of what it is you're presenting. So you need to sort of simplify it down. I mean, most of us have dashboards that are showing things like variant, diversity and so on in wastewater. sampling. And so that's sort of at a relatively simple level and easy to understand. But if you're going to try to get into some sort of dashboard that's actually going to start describing actual mutation analysis and so on, and then that will then prompt the question, well, what does this mean? And how are you going to describe that? And sometimes describing the ramifications, the biological ramifications of different mutations can be complicated and difficult for experts to actually agree on, let alone present on a public dashboard. So to present it to, and there was an interesting comment made yesterday about what data we report. Sometimes we separate, often we separate what we report to public health and academic partners or whoever compared to what we report to the public, because it just becomes too difficult to explain to the public. So I think that to get to a detailed level of sequence analysis reporting on a dashboard to the public starts to become almost prohibitively difficult. And it's not, I don't like saying that, because we also don't like to appear that we're trying to keep any secrets from the public. It's just, it can be very technically difficult to explain.
Go ahead, Prabhu.
Sure. I don't have as much experience with wastewater sequencing. It's still new for us, but I can try to extrapolate from the clinical side what I would like to see on the wastewater side, assuming that there's that same level of resolution. For me, I think it starts, or at least from the public, the so what? Why should they care about this? And then work backwards to figure out what you want to show. So right now, I think we still continue to show, the SARS-CoV-2 variant, figure, but I'm not sure that the public cares about that anymore, because the so what has been lost. But thinking back to SARS-CoV-2, there's specific examples. So I don't know if it's a dashboard as much as using it to answer questions. So this was, again, a couple of years ago. Our director was we were one of the more restrictive jurisdictions in terms of how we protected nursing homes. We, shut down group activities. We prevented visitors from coming in unless they could show a negative test and they were vaccinated. And there's getting a lot of pushback. And what we were able to show with sequencing was that in facilities that had prolonged outbreaks, there were multiple introductions. So we were able to look at different lineages and hypothesize that represented multiple introductions. And so we were able to show tangible, like, this is the reason why we need to maintain this. It's intuitive. It's, people know that the more people come in, the more likely you're going to have an outbreak, but we're able to show it tangibly in a way that helps support. So that's one use of it, is to be able to support policy. With hepatitis A right now, we had some preliminary trees, phylogenetic trees that we're looking at with the sequence cases, and it's been fascinating. It helped answer a lot of questions that we had on the public health side that we were not able to answer. One is, is primarily among people experiencing homelessness, but we start to see cases in people without risk factors. Is it spillover, or is these, two independent, sets of, chains? And we're able to show that clearly there's overlap, so there's, transmission between these populations. Is this, we have an outbreak in a nursing home, and trying to figure out where that came from, we're able to, through sequencing, be able to show actually it was potentially the result of infection control breach at a hospital. So there's all these specific questions that we had that we could look at, that we were able to answer, and that's what I think what we would highlight. I was fascinated to hear about the Hep A, that you're able to look at resolve specific cases in that wastewater. If you can get that level of resolution, I think there's a lot that we can do in wastewater that we're not able to do in the clinical sequencing side, just because, as you say, we don't have the depth of sampling that you need to be able to create those kind of trees. So, so just to, I guess, step back, it's so what, and I think the main value is to help us understand outbreak transmission dynamics and help us provide better context for understanding our surveillance data.
We have one more question, and that's all we have time for today. So I think that's going to be virtual.
All right, we had a question for the panelists from Gloria online. So in regards to broad/pathogen agnostic surveillance efforts, I was wondering if you've thought about any parallels between USAID's PREDICT project many years ago. It seemed to be a cautionary tale of seek and you will find. The effort stood down due to resource sustainability issues. Just curious if you've thought about it.
I'm not familiar with that project. So it's tough to answer that. Um, I'm not sure if there's a way to reframe that in a more general way.
I am also not familiar with the project.
Okay.
So maybe we can get back to this online question and pass it off to one here.
Okay, we have time for one more. Go ahead.
Perfect. I'm Tom Clerkin from UNC Chapel Hill, and this question is for Doris. In regards to those high KPC and oxa concentrations, have you started looking into like the different contributions of the biofilms and environmental contamination? Or how would you go about like designing a study to parse that out further?
Thank you for the questions. We do think about, so we haven't figured out whether it is from biofilm, but we do think about some further studies that we want to look at to determine whether it is from the environment from biofilm or from the facility itself. So one of the thought that we have is incorporating culture methods and also sequencing. So we know if we do sequencing, it will be amplifying everything in the wastewater, including those from the biofilm. But I think with culturing, it will really help us eliminate some of the possibility of the biofilm. And also, we do think about some of the more technical thing that we suggested to the nursing home, and we might implement it is one of the method is to disinfect the sewer pipeline in the facilities using Clorox foam. So this is a rather serious kind of project. So we do consult the CDC, and CDC recommended some of the products and cautious precaution that we have to do. So we have yet to apply this into our research. So one of the reasons is also we are short-handed. So when we have new fellows in the coming week, then we will be looking into the possibility of finding out where does the KPC and oxa 48 comes from in terms of wastewater detection.
Well, thank you very much. That's all we have time for. Please join me in thanking our panelists this morning.